All right, anatomy and physiology. Here is our lesson for today. I currently only have 5 minutes per video, so this may be broken into a sequence of different videos for you, and I'll put them in a playlist. So don't just watch the first 5 minutes, continue watching until we get through our content for today. I want you to do a bell ringer for today. Feel free to even pause this video now so that you can answer this question, identifying A through E, on your bell ringer sheet. Yes, I'm about to tell you the answers, but really pause. See if you can find things that look like those pictures in your notes. Hint, page 3 has, I think, 3 of them. Welcome back. I'm sure you all paused and didn't just wait for me to give you the answers. So A is referring to one of these guys, your monosaccharide. B is a big blob-looking thing, very similar to our globular or functional protein. Right, this guy, that's what B is. C is a long chain of A's, which are monosaccharides. Okay, so C would be your polysaccharides that you see here. D has 3 oxygens and 3 little tails, like fatty acid tails coming off of it. So that would be this guy here, your fat. Okay. It's not a phospholipid because there's no phosphate mentioned, and there's 3 tails instead of 2. Phospholipids have 2 tails. Then lastly, E is what we're actually going to talk about today. But if you flipped through your packet, okay, we're going to talk about this guy right here, which is going to be ATP. It has the nitrogenous base, it has a 5-carbon ring, and it also has a phosphate group right here. All right, well, let's talk about what do I mean when I'm talking about things. I'm getting ahead of myself, much like I do in class. Sorry. I wanted to recap the lab from yesterday for anybody that was absent. This slideshow is posted already in Google Classroom along with the Google form from yesterday, which was your only homework assignment to complete yesterday. But what we did was we wrote down this information in our lab notebooks. Even if you were absent yesterday, please be sure to write this down in your lab notebook. The data table I have provided for you, all you're going to write in the data table is the color change of each of the different solutions. What we did with Benedict's was we added this blue substance to water, glucose, sucrose, starch, and an unknown urine sample. And then we had to put it in a hot bath to warm it up in order to activate the indicator. And Benedict's changes from a blue to orange when it's indicating the thing it indicates. That was the point of the lab, to figure that out. Iodine, you didn't need to warm it up at all, but Mrs. Lloyd came around with some drops of kind of this brownish orangish substance, and it also indicates a certain substance. So we recorded our observations. What did these substances look like? What colors were they in the data table to see if there was any kind of change? For each of these that I took a picture of, I put it next to the control. This is water. And so water is not reactive at all. And so if it's different from the control, that indicates that Benedict's is testing for, as you see here, it goes from blue to orange, glucose. Benedict's does test for glucose. Sucrose, no change in the color. And so that tells me Benedict's does not test for sucrose. It also does not test for starch. But then students had their iodine test next. The control, again, this is just iodine and water, no reaction, and glucose, no reaction. Iodine does not test for glucose. No reaction here, does not test for sucrose. Here we do have a color change. Many students ended up with a little black spot kind of at the bottom of their dish if the starch had settled out of the water. But it changes to more of a brown, black, or bluish black color in iodine. So iodine does